brief on introductions of cytology. In content, introductions, definitions, classifications of cytology, advantage and disadvantage of diagnosis cytology. In introductions, cytopathologic diagnosis was discovered in 1920 by Dr. Jones and Pepino-Coulers. After the 10-year technicals of fine needle expression cytology was also introduced by Martin and Ellis and Stewart in United States. After some year, again in 15 January 1979, India Council of Medical Research established a permanent institute, the Cytology Research Center at Delhi. After that, what happened? Explorative cytology and fine needle cytologies are simple and invasive method for obtaining the tissue diagnosis of urinary tracts and other body cavities, and they study in the fields of cytologists. And come to the definition, we have to know it. What is cytology? Cytology is the science that it is the study of the cells, formation, structure, and functions, or the cells, the study of cells, study formations and its functions. And classification, it has two types of classification. One is the Norman and second one is the diagnosis cytology. Norman is deal with the Norman structure, formation and function of cell, and diagnosis cytology is deal with abnormal cells, structures and formations, which are further divided into two fronts. One is explorative cytology and interventional cytology. In explorative cytology, it is also known as the older branch that involve in the study of cells spontaneously switch off as epithelium surface, epithelial lining into the body cavity or body fluids. The cells for the study may be obtained from by scraping, brushings, and various mucosal surface. And the explorative cytology specimens are obtained from the female genital tract, respiratory tract, gastrointestinal tract, urinary tract, body fluids, other samples. For the female genital tract, the sample will collect from the lateral vaginal smear, vaginal pool smear, cervical smear. In respiratory tract, we collect from the sputum. For gastrointestinal tract, we collect from the endoscopic brushing. Urinary tract, we collect from the urinary Chandramani, blooded washings. For the body fluid, we will collect from the CSF and by synovial fluid. And other samples, we can do the buccal smears. And in the interventional cytology, it's mostly the samples are obtained from the clinical procedure or surgical intervention. It is mostly fine needle expressions, which also known as the expression biopsy cytology. And what happened in the interventional cytology is cytopathologists are often performed with the clinical procedure himself and interacted with the patients. And advantage of the diagnosis cytologists are their superior morphologists details will get, and the collections of the samples, speeds of the samples collections and diagnosis, and then no need to require for the surgeries or specialized anesthesia. And disadvantages are the limited information about structure, arrangement of cells within the lesions, and the other is less affinity diagnosed as the specific tumor by the distribution of an inflammatory. Thank you.